Hello students, after having a discussion on double fertilization, in today's lecture we are having a discussion on pollen-pistil interaction. Now what is pollen-pistil interaction? The word itself explains the meaning. It is the interaction between pollen grains and stigma. स्टिग्माला दुसरा वर्ड तुम्ही लिहू शकता स्पोरोफायटिक टिश्यू. Okay, so interaction between pollen grains and stigma is called as pollen-pistil interaction. It begins with pollination. And ends to fertilization. Okay. Now, in pollination, what we discussed, it is the transfer of pollen grains from anther to the stigma of the flower. Okay. Pollination does not guarantee the transfer of right type of pollen. Often, wrong type of pollen also can land on stigma. Wrong type of pollen means pollen grains from another species, from another plant, also gets planted onto the stigma. Now, this pistil has that ability to recognize the correct pollen. Pistil has which ability? It has ability to recognize the correct pollen. Okay, it is called as compatible pollen. That means pollen grains from another species or wrong pollens are not accepted by the pistil. It only accepts the pollen grains from the same species. That means correct pollen or compatible pollen. What this compatible pollen do then? Compatible pollen absorbs water and nutrients. Compatible pollen then absorbs water and nutrients from the surface of stigma. They germinates and produce pollen tube. Pollen tube. Okay. Compatible pollen grains then absorb water and nutrients from the stigma. They germinate and produce pollen tube. This pollen tube then grows through style. Okay, its growth through style is determined by specific chemicals. Okay, the stigmatic surface provides essential requirements for a successful germination which are absent in pollen. That means all the requirement of pollen grains are then satisfied through stigmatic surface. They provide all the essential requirements for further successful germination. Okay, the pollen tube is then passed through ovule and reaches the embryo sac. कुठे पोहोचते pollen tube? Embryo sac, where it enters in one of the synergids. And bursts, and two male gametes of the pollen tube becomes free. One of them unites with female gamete to form the diploid zygote, and another one unites with secondary nucleus to form triple or triploid fusion nucleus. Okay, this all the activity in all together in collectively we can call it as pollen-pistil interaction. Got it? After pollen-pistil interaction, our next topic for the discussion is development of endosperm. Development of endosperm. Now what is meant by development of endosperm? The triploid primary endosperm nucleus repeatedly divides mitotically to form nutritive tissue called as endosperm. बघा, पॉलिनेशन मध्ये आपण काय शिकलो? वन ऑफ द मेल गमेट युनाइट्स विथ सेकंडरी न्यूक्लियस टू फॉर्म ट्रिपल फ्यूजन न्यूक्लियस. ओके, that is your primary endosperm nucleus then repeatedly divides mitotically to form nutritive tissue called as endosperm. Now depending upon the mode of the development of endosperm, there are three types of endosperm are seen in plants, out of which first type is nuclear endosperm. Look at these diagrams. Now nuclear endosperm means what? It is the most common type of endosperm. During the formation of nuclear endosperm, the primary endosperm nucleus PEN, primary endosperm nucleus undergoes free nuclear divisions forming a large number of triploid nuclei which remain freely suspended in the common cytoplasm of cell. हे बघा, primary endosperm nucleus divides into large number of triploid nuclei which remain suspended in the common cytoplasm of cell. ओके, आता सेकंड डायग्राम मध्ये बघा, व्हॅक्युअल आहे हा. अ सेंट्रल व्हॅक्युअल पुशेस these all the nuclei toward the periphery. आता पेरिफेरी म्हणजे हा बघा, पेरिफेरी म्हणजे सर्कम्फरन्स, म्हणजे हा सेंट्रलचा व्हॅक्युअल ते जेवढे काही न्यूक्लिआय फॉर्म झाले त्याला कुठे सेंड करतो? इट पुशेस टुवर्ड द पेरिफेरी. लॅटर ऑन वॉल फॉर्मेशन टेक्स प्लेस. बघा, वॉल फॉर्मेशन टेक्स प्लेस टू फॉर्म अ सेल्युलर मास. हे बघा, हा एक सेल, दुसरा सेल, तिसरा सेल, अशा पद्धतीनं हे फ्री सेल होते. नंतरच्या स्टेजमध्ये काय झालं? वॉल इज फॉर्म टू फॉर्म इच सेल. या सगळ्यांना एकत्रितपणे वर्ड काय बोलाल तुम्ही? सेल्युलर मास. ओके, इट इज कॉमनली ऑब्झर्व्ड इन मेज, sunflower, etc. That's all about the nuclear type of endosperm. Then second endosperm type is cellular endosperm. Look at these diagrams. Cellular endosperm. The triploid primary endosperm nucleus undergoes nuclear division followed by cytokinesis. Followed by cytokinesis. Now what is meant by cytokinesis? During the division of nuclei, the cytoplasm also gets divided. That is called as cytokinesis. आता न्यूक्लियस पण डिवाइड होतो, सायटोप्लाझम पण डिवाइड होतो. Owing to this, the development of endosperm occurs in which form? न्यूक्लियस, त्याच्या बाजूला सायटोप्लाझम, त्यालाच काय बोलाल? सेल. दॅट मीन्स डिव्हिजन. दिस डिव्हिजन occurs in cellular form. Okay, it is less common and seen in dicot plants like datura, petunia. Then another example you can take balsam, etc. Okay, that's all about the cellular type of endosperm. Then third type of endosperm is halobial type of endosperm. What? Halobial type of endosperm. In halobial type of endosperm, the first division of PEN means what? Primary endosperm nucleus is followed by the formation of cell wall. Owing to this, the central cell is divided into a large micropylar cell and a small callosal cell. Micropylar cell and callosal cell. In both the cell, the further development of endosperm is of nuclear type. Wall develops between the nuclei in micropylar chamber. This type of embryo development is seen in plants belonging to order Halobiales. त्या प्लांटचा ऑर्डर काय आहे? हेलोबियल्स. हेलोबियल ऑर्डरच्या प्लांटमध्ये या टाईपचे एंडोस्पर्मची डेव्हलपमेंट तुम्हाला बघायला मिळते. फॉर एक्झाम्पल ॲस्फोडेइस. एक्झाम्पल काय घ्यायचं? ॲस्फोडेइस. In this way, that's all about the development of endosperm. In short, once again, depending on the mode of endosperm development, three types of endosperms are there. Nuclear endosperm, these are the diagrams. Then cellular endosperm. Then last one we discussed, halobial endosperm. Got it? Okay.